Well, you're most welcome to this talk. It's Sunday evening, the 30th of May. Now, there's been a lot of pessimism, even amongst sort of senior authority figures, about immunity to SARS coronavirus 2 after infection and vaccination not lasting for very long. And a lot of that was based on the idea that the seasonal coronaviruses, there's 4 seasonal coronaviruses that cause common cold symptoms, the immunity from those actually only does last for 6 to 12 months. But I think we're getting some much more optimistic data coming out now, indicating that my long-held view that there's likely to be long-lived immunity after SARS coronavirus 2 is starting to be supported by the data. Now, the first paper we want to look at briefly is this one. Duration of antibody response after severe acute respiratory syndrome. Now, this came out in 2007. So, of course, this is talking about SARS coronavirus 1. The pandemic, it was a pandemic. It spread to a few countries in 2003. Nothing like on the scale of this one, of course. So duration of antibody response after severe acute respiratory syndrome, 2007. 176 subjects. SARS-specific antibodies were maintained for an average of 2 years. Remember, this is SARS coronavirus 1 from the 2003 outbreak. Significant reduction in immunoglobulin G in the 3rd year. So from that, it's looking like the immunity lasts for about 2 years from SARS coronavirus 1 from the 2003 outbreak. So that was the first paper, and people have focused a lot on this to be pessimistic about the current outbreak. But this is from the British Medical Journal, so this is the next paper we're looking at here. That's this one. Do many people have pre-existing immunity? Quite a fascinating article, actually. It talks about a whole range of things related to immunity. Well worth a read. The link is there. Just click on it. But the thing I wanted to point out from this article is that it's, as well as talking about the antibody response, it's talking about the cellular response. But T-cell studies allow for a substantially different, more optimistic interpretation compared to just looking at antibodies, is what this this paper talks about. So let's go on and look specifically now at one of these papers, and that's this one. So in this paper here, so let's look at this one now. Now this actually came out in 2020. SARS coronavirus 2 specific T-cell mediated antibodies. Now remember, these T cells are the T lymphocytes. Just a quick revise. We've done this quite a few times, but the white cells, the protective cells, are the lymphocytes. One type of white cell, the lymphocytes. And there's 2 sorts. There's the large and there's the small. The large are the natural killer cells that we've looked at before that kill virally infected cells. The small are divided into the B and the T lymphocytes. So we're talking about the T lymphocytes here. So the small lymphocytes are the B and the T lymphocytes. And the T are important because there's T helpers, T suppressors and T cytotoxic cells. So it's the T helper cells that help the B cells to make the antibodies. That's the way it works. So we're talking about these T cells, that they're a type of white blood cell, a type of leukocyte. Anyway, back to the paper. Now, only 36 patients involved in this, but published in Nature in 2020. Now, in these 36 patients, in all of these individuals, we found CD4 and CD8 cells that recognize multiple regions of the N protein. That's a bit complicated already. But the CD4 and CD8 are both types of T lymphocyte. Both types of T lymphocyte. The CD4 are actually the T helper cells, and the CD8 are actually the T cytotoxic cells. But anyway, in patients that have been infected with SARS coronavirus 2, they found these cells that were activated to multiple regions of the N protein, the nucleoplasmid protein. More on that in a minute. Coronavirus nucleoplasmid N structure protein that forms complexes with genomic RNA. So the genomic RNA is the RNA of the virus. So basically what you've got with the SARS coronavirus is you've got the virus there, and you've got your strand of RNA in there, and then you've got your spike proteins there. So the nucleoplasmid proteins are associated with the RNA in the nucleus. Well, it's not really a nucleus, it's the centre of the cell. Then these are the spike proteins here, and these are the membrane proteins in the membrane roundabout there. So basically we've got these three lots of proteins: spike protein, membrane proteins, and then these internal ones are the nucleoplasmid proteins. Now, studies could look at any of these, but different studies tend to look at different ones because it's easier to analyse one particular aspect of the immune response. But these are all immune responses generated to the antigens, which are the virus, whether it's the N antigens, the S antigens, or the M antigens, the nucleoplasmid, the spike, or the membrane antigens. They're all parts of the virus that your immune system recognises as being foreign. And of course, the immune system is just the most mind-blowing system. It's quite incredible. Anyway, these 36 patients, so coronavirus nucleoplasmid structural protein, so we said that. Patients, so what they found is that all 36 patients, all of these 36 patients, in the months after infection, had these activated T cells, the CD4 helper cells that will help the B cells to make the antibodies, and the CD8 T cells that will directly kill virally infected cells. In fact, I've got a diagram here from my physiology notes book. So these are actual, I've actually labelled these as natural killer cells, but the same would apply to T cytotoxic lymphocytes. So they can directly kill sometimes cancer cells, and they can directly kill virally infected cells. So this natural killer cell, or the T cytotoxic cell, they work in the same way, comes next to the virally infected cell, drops these proteins called perforins on it, and these perforate the cell, let water into the cell, and this cell is basically killed. And as that cell is killed, the viruses are killed with it. So that's the way the T cytotoxic and the NK cells are working. But anyway, so they found these cells were there, and they got rid of the virus. They were effective, and they were still there after the infection. But then, and this is the most encouraging thing I'm going to tell you today, is remarkably encouraging. They found 23 patients. This is a study done in Singapore. So to find patients, 36 patients that they're able to analyse that were infected with the SARS coronavirus 2. No big deal, we've got plenty of those. But then they found out 23 patients who'd suffered from SARS, severe acute respiratory syndrome, in 2003 with the SARS coronavirus 1 infection. So these are people that were infected with a similar virus. This study was done in 2020, so that was 17 years before that. 23 patients possessed long-lasting memory T cells that were still reactive. 17 years later, the infection was in 2003, the tests were done in 2020, and these cells, the helper cells and the killer cells, were still there. So SARS coronavirus 1 now no longer exists in the world, but if these patients were reinfected with it, they would still be immune to it, or have a large level of immunity to it 17 years later. So that's the paper there. Don't take my word for it. Read it for yourself. It's all there, and a very interesting read, if a bit detailed in places. Right, I'm back on the right screen. Yeah, these T cells display robust cross-reactivity to the nucleoplasmic protein of SARS coronavirus 2. In other words, people that have had SARS coronavirus 1 are likely to have a large degree of immunity to SARS coronavirus 2, cross-reactivity. Now, I think from memory there was only about 80,000 people had the infection in 2003 because it was contained. But those 80,000 people would be immune to SARS coronavirus 2, or largely immune to SARS coronavirus 2, 17 years later on. This is why I'm optimistic that the immunity we're now generating to SARS coronavirus 2 is going to be long-lived. But more on that in a minute when I give you the precise evidence for that. Epitope characteristic of specific T cells shows the recognition of protein fragments that are conserved amongst animal beta coronaviruses. Now, of course, SARS coronavirus 2 is a beta type coronavirus. Now, the epitope is the part of the virus that the immune system recognises as being foreign. It's the antigen, the epitope. And what this is saying is that these cells, these immune cells that were generated in response to SARS coronavirus 1, recognise protein fragments on many different types. When it says it's conserved, that's an evolutionary term. It means it's been conserved through viral evolution. And it means that it occurs in many different animal coronaviruses. So presumably out there in the animal kingdom, there's untold, hundreds of different types of coronaviruses. What this is saying is that the immunity from SARS coronavirus 1 will extend against a wide range of those. But they are different to the common cold coronaviruses that cause the common cold. So the animal coronaviruses that we have at the moment are the SARS coronavirus 1, well, it's not there anymore, but the SARS coronavirus 2, and of course the Middle East respiratory syndrome virus are the three. But were others to jump from the animal kingdom to humans, or however it got here, then it's presumed that there will be a degree of immunity in those people that suffer from SARS coronavirus 1, because the proteins are highly conserved across different viruses in the animal coronavirus groupings, populations of coronaviruses. So next paper, persistent cellular immunity. Now this one actually is a preprint. So that is this one here. But it looks like a pretty well written paper. So I'm going to tell you about it anyway. I've read it. I'm not a good peer reviewer, but it looks okay to me. Now, this is persistent cellular immunity in SARS coronavirus infection. And this is looking particularly at T cell immunity, this particular paper. So cellular and hormonal immune response to infection. Now, what this means is when you have SARS coronavirus 2, you're going to develop a cellular response. That's the lymphocytes, the B and the T cells. That's the cellular immunity. But you also develop a humoral immunity. Now, humours relate to the fluids in the body. So we talk about being in good humour. That goes on old theories relating to fluids in the body. But humours still mean related to the fluids in the body. So humoral immunity is the immunity in the fluids of the body, that is in the antibodies that are circulating throughout the blood and the tissue fluids. So in other words, the infection with SARS coronavirus 2 causes a cellular response and a humoral response, an antibody response. That's what that means. Antibody responses include most of the structural proteins expressed by the virus. So we know that there's different, as we've said, there's different proteins on the virus. There's the nucleoplasmid, there's the spike, there's the membrane. Antibodies to all of these are made. Specific antibodies that fit the reciprocal shape of the specific proteins that make up the virus. So antibody responses include most of the structural proteins expressed. So it's a polyclonal antibody response in response to the different components of the proteome of the virus. Neutralizing antibodies directed primarily to the receptor binding domain of the spike protein. So we know those neutralizing antibodies that they fit onto the spike protein, and when the antibody fits onto the spike protein, so if that's the spike protein there, and there's some antibody sticking onto it like that, then that means it can't fit into its receptor site. The antibody gets in the way. Whereas if the antibody's not there, the neutralizing antibody's not there, it fits in and you get the infection. So the neutralizing antibodies mostly affecting the spike protein, but there's other antibodies as well. And what these other antibodies will do is they will actually agglutinate the viral particles. So they will clump a lot of them all together because the antibodies are sticky and they'll clump a lot of the viruses together. You'll end up with a clump of hundreds or thousands of viruses all clumped together. And then one of these big cells that eat fragments, one of the phagocytes, one of the macrophages can come along and eat it all together. It's said that antibodies prepare the food for the phagocytes table, the eating cells. So it clumps them all together. So two ways the antibodies are getting rid of it. But moving on, neutralizing antibodies, I said that. But lymphopenia is a prominent feature of severe infection. Now, lymphocytosis would be an increase in the number of lymphocytes in the blood. A lymphopenia would be a reduction in the amount of lymphocytes in the blood. So when people have severe COVID, severe infection caused by SARS coronavirus 2, they get a lymphopenia. The number of lymphocytes goes down. Now, it's the lymphocytes that are combating the infection, so you'd expect the number of lymphocytes to go up. But actually what happens is these blood tests that measure a lymphopenia, a reduction in the number of lymphocytes in the blood, they're measuring just that, the number of lymphocytes in the blood. But because the lymphocytes are needed in the tissues to kill the infection, the lymphocytes move from the blood into the tissues. Therefore, the number of lymphocytes in the blood goes down. It's quite clever, really. So if people have a lymphopenia during a viral infection, that's an indicator that these lymphocytes are essential for fighting the viral infection because they moved into the tissues to do their job, to do the business of getting rid of the virus. So during acute infection, T cells display highly activated cytotoxic phenotype. This is just a scientific way of saying that the form of the cells and the function of the cells is to be cytotoxic and to kill virally infected cells, as we showed in that diagram there. Virally infected cells are eliminated. These cells develop a phenotype, a way of a structure and a function which is precise to killing cells, which of course is exactly what we want, then the virus can't spread around the body. A convalescent plasma harbours polyfunctional SARS coronavirus 2 T cells. So when people get better, they have many functional T cells that have got rid of this virus and they remain in the blood for a while. But then these cells often display a stem-like memory phenotype. Again, phenotype is the structure and function that you actually see. Stem-like means it's related to stem cells. So related to stem cells, and stem cells are any cell which is capable of further differentiation. Now, I know it's getting a bit complicated today, but I know quite a lot of you like this kind of stuff. So let's just look at this in a little more detail. So this is from my physiology book. So here we see that this particular lymphocyte has detected, this is a bacteria, of course, the virus will be way smaller. Now what happens is that the particular lymphocyte here that is detected, that is correct for this particular type of bacteria, or in the case of SARS coronavirus 2, of course, a virus, then what happens is this cell divides, and it divides into two populations. This population here divides and divides again and divides again and divides again and divides again to make a huge population of these, and these cells in this case are B cells, but these are plasma cells, billions of them, and these produce the antibodies. But we get the same thing with T cells. We get proliferation of T cells as well. And whether it's B cells or T cells, this cell also divides into another cell here. And this cell here remembers exactly how to combat that virus or bacteria, but it's not functional. But it does divide, and it divides into a group of cells like this. And this group of cells here, these are the memory cells. They remember how to attack this. And then if the infection comes along again, some of these can divide again into more that produce a population of the effector cells. So we have these memory stem type cells in the B and T response. The immune system is absolutely incredible. And these have cross-reactivity with seasonal common cold coronaviruses. Now, why is it that some people get severe infection with SARS coronavirus 2 and other people get less severe infection? Well, we know about all the risk factors and the comorbidities. But even so, you can get someone with the same comorbidities, the same age, same sex. One gets bad disease, one doesn't get bad disease. The answer may be that the one that doesn't get as bad disease had infection with the seasonal coronavirus 3 months ago, had a common cold with it, and had a degree of immunity because they were still within that 6 to 12 months where the immunity lasts after the SARS, after the common cold coronaviruses. So cross-reactivity could be a factor in determining that, and the authors do say it could be associated with a milder course of disease. Coronavirus levels persistent immunity. Now, for seasonal coronaviruses, the immunity there is only lasting for 6 to 12 months, we know that. But we've seen that with SARS coronavirus, one, it lasts for 17 years. Immune response is, in recovered individuals, they can measure the T cell response. So that's what they did in this study. And they had paired samples of people. They looked at them 1.3 months after the infection and 6.1 months after the infection. So they're able to compare the infection 1.3 months after the infection and 6.1 months after the infection. 41 people, males and females, variety of ages, various severities of disease, but most of the diseases were fairly mild. And recovered individuals shows persistent polyfunctional SARS coronavirus 2 antigen-specific memory. So they were able to see these memory cells in the blood. And that's important to notice that at this stage they were seeing these cells in the blood. And that could contribute to a rapid recall response if they were reinfected. In other words, it would give immunity in recovered individuals. So enduring immune alteration numbers in the blood of CD4 helper cells, CD8 T cells, cytotoxic cells in the blood up to 6 months after infection. So it's looking good for 6 months. Now, summary here is broadly reactive and highly functional memory T cell response. Yes, that occurred. Persisted 6 months after the infection. We know that, but of course we haven't had too much time to work it all out yet. Enduring immune alterations in CD4 and CD8 cell compartments. In other words, this is talking about the memory compartment. So there were memory cells that they detected in the blood. But specific circulating T cell numbers goes down over 6 months. So the amounts of these specific T cells that particularly fight the SARS coronavirus 2 does go down after 6 months. Does this mean that the infection might only last for 6 months? The answer is no, because turns out that these memory cells only stay in the blood for a few months. Then these memory cells migrate into the bone marrow. They migrate into the bone marrow and they stay in the bone marrow. And if they're living in the bone marrow, of course, you can't detect them in the blood. Now, there's two sorts of bone marrow. There's yellow bone marrow and red bone marrow. The long bones, like your femur and your humerus and your radius and your ulna, they contain mostly yellow bone marrow, especially in the middle. They do have some red bone marrow at the epiphyses at the end. Flat bones like your sternum, your skull, your pelvis, very rich in red bone marrow. So that is where these cells reside. But how do you find out if these cells are in the bone marrow? Well, the only way to do that is to stick, and you often do it through the sternum, stick a needle through there and suck out some of the bone marrow. So a very invasive, painful procedure. Well, it depends who does it. Local anaesthetic, but you know, it's a traumatic procedure. You know, if someone says you need a blood sample, then that's one thing. But if you're going to stick something into your bone marrow, that's something else. So that's why this piece of research is particularly appreciated and impressive. And this is SARS coronavirus 2 infection induced long-lived bone marrow plasma cells in humans. Now, the plasma cells are the B cells, the B lymphocytes. And of course, the only way they know this is because noble people, and there's no other way to put it, noble people allowed their bone marrow to be sampled. So the one we were looking at before was T cell response. We've seen that has longevity in the blood, but that's limited. Now, this paper here is not looking for the presence of T cells in the bone marrow. If it looked for them, I suspect it would find them. But we can only report what the paper's saying. It was looking for the presence of long-lived plasma cells, which are the B cells that actually make the antibodies. So this particular study was only actually looking for the presence of the B cells, the B lymphocytes. But that's what we've got. So, but of course, that's part of it. It would be very surprising if the B cells persisted and the T didn't, but it's just what they were looking for. So we can only say what it says, but it's very encouraging. Long-lived bone marrow plasma memory cells were found in the bone marrow. Convalescent individuals have a significantly lower risk of reinfection. We know that. Patients who experience mild infection, there were 77 of them. Spike antibodies decline rapidly in the first 4 months after infection, then more gradually over the following 7 months. So the amount of antibodies in the blood are going down fairly quickly for 4 months and then going down more slowly for the next 7 months. But the time you get to about 11 months, though, the numbers have dropped off quite significantly and they can be hard to detect. So that's looking bad. So spike protein antibodies decline rapidly over the first 4 months, then more gradually over the next 7 months, remaining detectable at least 11 months after the infection, but sometimes a year after the infection you might not detect them, depending on how sensitive your detection is. So this looks pessimistic, but actually it's not, because if you think about it, today I've probably been exposed to 30 or 40 viruses, a dozen types of bacteria. So every day we're exposed to these viruses. And we're making antibodies. Most of the time we don't get sick because we make the immune response before we get sick. But if we had antibodies circulating in our blood to all of the viruses and all of the bacteria we'd ever been exposed to in our life, then our blood would be thick sludge and we'd all die of thrombosis or something. So you can't keep high levels of antibodies for everything in the blood. That would be ridiculous. So it's not surprising. In fact, I would 100% expect the antibodies to go down. It's exactly what you'd expect. But bone marrow aspirate, and they were looking at spike-specific bone marrow plasma cells. So looking at one particular type of, in other words, the plasma cells that knew how to make the antibodies to the spike protein is what they're looking at. You can't look at everything in one study. That's what they looked at. 18 SARS coronavirus 2 convalescent patients, 7 to 8 months after infection. So 7 to 8 months after the infection, they're looking at the bone marrow. 5 patients, they looked at 11 months after the infection. So they're looking at patients 7 to 8 months and 11 months after the infection. Now, some patients were vaccinated towards the end of this experiment, so that they dropped out. This is looking at natural immunity. It's SARS coronavirus 2 infection we're looking at here. So 7 to 8 months and then 5 patients at 11 months. Numbers don't sound good, but if they all have it, then that's pretty promising, and they did. So 7 to 8 months, there was no plasma blast cells. Blast cells are cells that produce something. In this case, they produce antibodies. So 7 to 8 months, there was no specific B lymphocytes. That's no specific B lymphocytes, blast cells that make the antibodies in the plasma. They weren't in the plasma anymore. There were still some antibodies that lingered on, because antibodies can linger on for a few months, but the cells weren't there. But they did find the cells in the bone marrow. Now, as well as that, here they had 11 patients who had not had the infection, who also volunteered for bone marrow samples, and none of those had these cells. But the ones who, these patients here, 7 to 8 months after infection, and a few patients 11 months after infection, they had these specific bone marrow plasma cells. In other words, these B lymphocytes. They had that in their bone marrow, just waiting in case it had to do its job again. They were there, waiting in the bone marrow with the memory of exactly how to combat this virus. Quite amazing, the immune system. We demonstrated, direct quotes, we demonstrated that S-binding bone marrow plasma cells, in other words, the B lymphocytes, are quiescent. They're sitting there quietly, indicating that they are part of a long-lived compartment, a long-lived immune compartment in the bone marrow. Overall, we show that SARS coronavirus 2 infection induces a robust antigen-specific, long-lived humoral response in humans, because these cells can make the antibodies that go into the body humours, into the fluids, and will confer immunity. And because it's true of the B cells, I would imagine if they looked, it would be true of the T cells as well. It would be really quite unimaginable that you would have memory B cells without memory T cells. In fact, really you must have, because it's the T cells that tell the B cells how to make the antibodies, so you must have. They must be there, it's just that this study didn't look for them specifically. So 7 to 8 months and 11 months after infection with SARS coronavirus 2, the cells are there in the bone marrow, and no reason why they shouldn't go away. They can just sit there in the bone marrow for years, as the cells in SARS coronavirus 2 sat there for 17 years. That's why I'm optimistic that we're going to get rid of this virus. Right, just a couple of words from the pundits before we finish. Scott Hennessy, immunologist, University of Pennsylvania. The papers are consistent with a growing body of literature that suggests that immunity elicited by infection and vaccination for SARS coronavirus 2 appears to be long-lived. So he's happy that this will apply to vaccination-induced immunity as well as infection-induced immunity. What seems to be particularly effective is the infection followed by the vaccine. That seems to be a particularly good immune response. The reason we get infected with common coronaviruses repetitively, that's the common cold coronavirus, just throughout life, might have much more to do with the variation of these viruses rather than the immunity. In other words, the common cold coronaviruses mutate much quicker than SARS coronavirus 1 and SARS coronavirus 2, notwithstanding the mutants that we have. That is still a true statement. Immunologist Rockefeller, New York. People who are infected and get vaccinated really have a terrific response, a terrific set of antibodies, because they continue to evolve their antibodies, because the cells are hiding in the bone marrow and can continue to make antibodies. I expect that they will last for a long time. Long time to me. Don't want to put words in his mouth, but we're talking years. A booster dose is going to be necessary, probably for those who've only been vaccinated, so they might need a third dose to get their immunity up. But people who've had the infection and then had the vaccine will probably be immune for, I think, for a decade or 2 or 3. But we're going to know about a third dose pretty soon. Will a third dose give immunity which is comparable to natural immunity? Well, with other vaccines, it does. Right, if you watched that, well done, because I know that's quite scientific, but let me just give you a quick summary. Because the immunity to SARS coronavirus 1 is long-lived, and I think we've given evidence now that the immunity to SARS coronavirus 2 is also going to be long-lived. As more and more people become immune, I think this infection can be eradicated from the world, providing we get vaccines out to everyone that needs it. Or additional vaccines to the people, or one additional vaccine perhaps to people that have already been infected. So cause for optimism in the medium term. It's going to take a few years, a few seasons to get rid of it. Now, I might be wrong. Some senior doctors in the UK have said this is going to become endemic indefinitely. But I think the evidence is starting to accumulate in support of my position that we can get rid of this virus after a few seasons. Don't know how many, but a few seasons. So I am hopeful for that. And if you watched this video all the way through, thank you for watching. We'll have an easier one next time, a news one. Oh, and I've got a report from Susan in Australia for the next video as well. So tune in for that. Thank you for watching.